Hello everyone. So today we'll be looking at autoimmunity and hypersensitivity reactions. So we'll be looking at quite a few different autoimmune disorders and go through our type 1 to 4 hypersensitivity reactions, as well as some methods of tolerance and how autoimmunity is prevented. So again, autoimmunity may be induced due to the failure of tolerance, which we'll get into, mechanisms and or environmental factors such as infection. So autoimmune disease. Mechanisms of damage in autoimmune diseases often involve type 2, 3, and 4 hypersensitive reactions. This damage is often due to self-reactive antibodies and complement actions. These also include our CDLs, Th1 T cells, and our Th17 cells. So remember, our Th1s are involved in cell-mediated immunity, and our Th17 are involved with pro-inflammatory. So here's a summary of that here. So we have different categories of autoimmune disease. We have organ-specific autoimmunity and systemic autoimmunity. If you go to the Janway's textbook, chapter 15 is on this, and chapter 14 as well. So we'll go through some of these in more detail, but here's essentially the main list of all the different diseases and mechanisms. So again, we've probably heard of a lot of these, such as psoriasis, rheumatoid arthritis, Graves' disease, Hashimoto's thyroiditis, systemic lupus erythematosus, Sjogren's disease, Crohn's disease, multiple sclerosis, and type 1 diabetes. So again, here are the different mechanisms. So autoreactive, you can see autoreactive is essentially in every single one. We either have autoantibodies or autoreactive T cells. So for example, in diabetes, against our beta cells and our pancreatic islets, you can see your beta cells, leading to lack of insulin production. And if you want more detail on these, you can check it out, but we'll go through some of them in detail. Again, so here are layers of tolerance. So these are what prevents autoimmunity. So we have central tolerance, antigen segregation, peripheral anergy, which you talked about anergy before, our regulatory T cells are our Tregs, and our functional deviation and activation-induced cell death. So again, here we can see the different sets of actions. So central tolerance occurs in our primary lymphoid organs, in the thymus and bone marrow. Antigen segregation, our peripheral organs, thyroid and pancreas, for example. Peripheral energy in our secondary lymphoid tissues, and etc. And we'll again go through these in more detail. So there are several mechanisms work together to prevent autoimmune reactions. So again, central tolerance involves the deletion of self-reactive T cells and B cells. So T cells, we talked about our positive and negative selection, and we also had similar mechanisms seen in our B cells. So individual organs of the body express certain tissue-specific antigens. And we also mentioned the AR molecule that presented self molecules to the B cells and T cells, essentially to develop tolerance to them. So in the thymus, T cells arise capable of recognizing tissue-specific antigens under the control of our AIRE protein. Thymic medullary cells express tissue-specific proteins, leading to the deletion of tissue-reactive T cells. In the absence of AIRE, T cells reactive to tissue-specific antigens mature and leave the thymus, which is not good. So central tolerance is essentially central deletion of autoreactive T cells. The central deletion or inactivation of newly formed lymphocytes is the first checkpoint of self-tolerance. So again, we mentioned our T regulatory cells, which are extremely important. So these are T cells. These T regulatory cells produce cytokines, like we talked about, TGF beta and IL-10. So let's write that down. So like TGF, beta, and IL-10. And these are important in maintaining tolerance and preventing autoimmune reactions. So as we said, we have central tolerance, which is the deletion of self-reactive T and B cells. We also have peripheral tolerance, where we have clonal anergy of T cells due to the lack of a secondary signal. So that secondary signal, again, we talked about the CD40 ligand or the binding of CD28 to the B7 molecules. So again, here's an example of our regulatory tolerance from Tregs. So we have a T cell specific for self-antigen recognized in the thymus, becomes a natural Treg or N-Treg. T cell specific for self or commensal microbiota antigen is recognized in the presence of TGF-beta and becomes an induced T regulatory cell. So there's also two subsets of Tregs, the N-Treg and the I-Treg. And again, these cytokines IL-10 and TGF-beta are produced by Treg cells and inhibit other self-reactive cells, essentially given that negative symbol. And this occurs in the periphery. So Tregs respond to self-antigens by reducing inhibitory cytokines, IL-10 and TGF-beta, and these shut down the activity of other T cells. So in a lot of studies, they'll look at pro versus anti-inflammatory, and a lot of studies will look at these two cytokines as the anti-inflammatory. Essentially, this just downregulates our responses. So in a lot of diseases, we have chronic inflammation. So sometimes they look at exercise. Does exercise increase IL-10 and actually downregulate inflammation? So stuff like that. Or certain medications and so on. So again, peripheral energy involves a cellular inactivation by weak signaling without co-stimulus, and this occurs in our secondary lymphoid tissue. So there's consequences of this insufficient signaling. So we've seen this figure before. So antigen recognition in the absence of co-stimulation leads to functional inactivation or clonal deletion of peripheral T cells. So here we see in a normal T cell, we have a co-stimulator. Here we do not, and this T cell becomes anergic. And if we only have the binding of the co-stimulator with only single 2, we never had our CD4 and our MHC bind, then we also don't have an effect on the T cell. So if the T cell is the only signal delivered, we have anergy. The T cell becomes non-responsive to further stimulatory signals delivered through the T cell receptor. Anergy is a regulatory process to prevent inappropriate T cell activation and involves incomplete signaling as a mechanism to induce T cell tolerance. Again, we also have activation-induced cell death, causing apoptosis, and this occurs in the secondary lymphoid tissue and sites of inflammation. So again, we have our central tolerance and our peripheral tolerance. We can also have activation-induced cell death of autoreactive B cells after somatic hypermutation. So we talked about somatic hypermutation, how they can rearrange to become the most effective cells. So again, somatic hypermutation generates novel B cell specificities within the germinal centers and the lymph nodes, and some of these B cells may be able to bind to self-antigens. And an encounter of autoreactive B cell with a self-antigen with the germinal center causes apoptosis. So again, we have those cells like an AIRE presenting antigens. This one recognizes itself, it's targeted to death. So we also have antigen segregation in certain sites. So these are sites that have limited lymphocyte access and have high TGF-beta production, which again was similar to our Tregs. So these down-regulate our activity. So these include our brain, for obvious reasons, eyes, testes, and uterus. So for example, a lot of people can get eye transplantation without any autoimmunity or transplantation reactions based on the fact that it's an immune-privileged site. So essentially, there's not going to be reactions going on there. Oops, sorry. So antigens and immunologically privileged sites do not induce immune attack, but can serve as targets. So damage to a site where self-antigens are hidden, such as the eye, can cause a release of hidden antigens into the tissues and can be processed and presented to T cells. Activated T cells can attack and attack the damaged eye and may infiltrate and attack healthy eye. So essentially what this is saying is that, so this is an immune-privileged site, so we can see here versus this side. So let's just say you have a traumatic accident, trauma to one eye. This can essentially allow immune cells to get in and receive antigens that are normally not exposed to the immune system, and essentially they'll be recognized as foreign. And unfortunately, this actually activates the other eye as well because these antigens are located in both. And so effector T cells can return via the bloodstream and encounter the antigen in both eyes and cause damage. So here's an example of sympathetic ophthalmia, which the physical damage to one eye ultimately induces damage to the other eye due to the activation of autoreactive T cells to sequestered antigens. And this can happen in other conditions as well, to other sites of the body as well. So, autoimmune disease can involve many actions. So, we can see here a list which involve many aspects of the immune system. So, example, SLE, T cells can be pathogenic and help for antibody. B cells present antigen to T cells antibodies pathogenic. Saying type 1 type 1 diabetes can be pathogenic for T cells. The B cells present antigen to the T cells, and the antibody is present but has an unclear role. An example we can see from myasthenia gravis and multiple sclerosis. So autoimmune diseases can be classified. So again, we talked about organ-specific and systemic autoimmune diseases. So these are ones that can affect pretty much the entire system. And then these are organ-specific. So again, myasthenia gravis would be in the nervous system. Multiple sclerosis, again, nervous system. Crohn's disease in the intestinal system, and psoriasis in the skin, and then diabetes in the pancreas. But they're often grouped into two categories, like we said, organ, systemic. So that's the main point here. So again, here's pretty much a nearly inclusive list of all the autoimmune disorders. So again, here are the different targets. So we can see you can pretty much target most things in our body. So we can see one here randomly. So again, testes were immune privilege. Somehow our immune cells get in. We can have spontaneous male infertility, where our immune cells target the spermatozoa. Same with the ovaries. We can have premature menopause if we target the corpus luteum. And again, here's a million. There's a ton. So again, you've probably heard of pernicious anemia with vitamin B12, and myasthenia gravis with the acetylcholine receptor, and etc. So again, if you want to go through this, if you're interested in any of these diseases, you can take a closer look. So now we're moving more into the hypersensitive reactions. So as we talked about, there are many different types. We have our type 1, 2, 3, and 4. And we had our immediate, intermediate, and delayed reactions. So this is a really good summary figure. If you want to come back to this, I recommend it, but we'll go through them in detail. So we've talked a bit about type 1, or type 4 and type 1, with type 4 in Th1 cells in our Th1 lecture, and type 2, type 1 responses in our Th2 lecture. So I recommend you check that out, but we'll take a look at type 2. So type 2 hypersensitive reactions, these have an intermediate timing. So we can see here that usually start within hours and may resolve within 24 hours. The mechanism is through antibodies, so that's kind of like the key thing, is that antibody causes damage to tissues or cells through complement fixation, opsonization, and antibody-dependent cellular cytotoxicity. So essentially, as you can see, these are all the functions of antibody. So essentially, we have our antibodies binding to a tissue that we do not want, and then again, causes all the normal functions of ADCC, opsonization, and complement fixation, which is not good. So, and again, type 2 hypersensitivity reactions, we have antibodies against cell surface proteins. So like an antibody, like IgM or IgE, causes damage to tissues or cells through complement fixation, opsonization, and ADCC. This attacks cells of the innate immune system. And then they are attacked by innate systems. So our NK cells, macrophages, neutrophils, eosinophils, all release their mediators of lytic enzymes, including TNF and ADCC, targeting their own cells. We also have our complement fixation, causing membrane attack complex, which causes damage to cell membranes. And then we have small C fragments, such as C5a, which act as chemoattractants. So it's essentially turning all the good things about complement and our antibody mediated responses against their cells, which is not good. And these can be quite powerful responses. As you can see, some of these are quite severe diseases that are involved in type 2 hypersensitive reactions. So again, key is antibody against cell surface proteins. So here we can see the syndromes. So hemolytic anemia, which is quite dangerous. Rh blood group antigens. Then we have destruction of our RBCs. We can have good Pasture disease, which attacks our non-collagenous domains of the basement membrane of collagen type 4. Cause glomerulonephritis and pulmonary hemorrhage, which are not good. And then acute rheumatic fever, causing arthritis, myocarditis, and late scarring of the heart valves. So we've seen type 2 reactions, you've probably heard of these reactions, with our blood antigens, with our 4 different types of A, B, O, and AB. So again, type A blood have antigen A on RBCs, but have anti-B IgM. Type B have antigen B antigen, and have anti-A, where a type O doesn't have A or B, and has IgM against both A and B, where type AB does not have antibodies and is against A or B. So that's the universal recipient and the universal donor. Again, here you can see the structures. And then here is kind of summary. So glutenins is what it's, and then isohemagglutinin. So our antibodies, so A anti-B, B anti-A, AB is none, and then type O is anti-A and anti-B. So what happens if the wrong blood group is received in a transfusion? So for example, a type O person receives type A anti-red blood cells. Their anti-A IgM will bind to this A antigen on the red blood cells, and again cause all the reactions just talked about with complement fixation, opsonization, and ADCC. This will cause red blood cell lysis. Then there will be the release of the free hemoglobin from these RBCs, which are then converted to bilirubin, which you may have learned in physiology about the breakdown of red blood cells. And this can result in rapid transfusion with fever and chills. If memory B cells produce IgG, there's delayed transfusion reaction following the lysis of any remaining A-positive red blood cells, which is quite bad. So this is summarized in this figure if you want to take another look. So another example is hemolytic disease of the newborn, erythroblastoma fetalis, where there's an anti-RH reaction against the fetus. So an erythroblastoma fetalis, or hemolytic disease of the newborn, occurs due to RH incompatibility between the mother and fetus. So remember, we have a RH positive and RH negative. So if someone says they're A positive, they're RH positive. So red blood cells can be Rh negative or Rh positive. So in hemolytic disease, an Rh negative individual responds to Rh positive red blood cells and makes antibodies against the Rh antigen, and this results in destruction of the Rh positive. So if an Rh negative mother has an Rh positive baby in her first pregnancy, this will have sensitization during the first pregnancy, and there's usually no complications. But in subsequent pregnancy, if there is an Rh positive child, there will be destruction of the fetal red blood cells by maternal antibody, IgG. So as we discussed, IgG can cross in the placenta, as we see here, and this results in complement fixation, opsonization, and ADCC by fetal macrophages. So this condition is essentially now can be prevented by passive immunization. So we treat the mother with an anti-Rh antibody during the first pregnancy. This is usually called RhoGAM. So that's the antibody that they give, and this prevents B cell sensitization and memory cell formation and causes no response to the Rh positive antibodies, or red blood cells, sorry. So here we see the RhoGAM. So another one we'll take a look at is good pastures. So in this scenario, we have antibody against matrix antigens. So remember, we had our collagen type 4. So in good pasture syndrome, we have autoreactive antibodies against alpha-3 chains of type 4 collagen in the basement membranes. This results in anti-collagen IgG antibody that binds to collagen in the basement membranes of the renal glomeruli and pulmonary alveoli in some cases, and this results in damage to the basement membrane. So here you can see a normal compared to the damaged. So in here, we have the damage. So good pasture disease, we have the deposition of IgG against type 4 collagen. This causes tissue damage mediated by complement and neutrophils. This can be rapidly fatal if untreated, which is quite serious. So we can see in good pastures that the bound anti-collagen antibodies interact with the Fc receptor on monocytes, neutrophils, basophils, and mast cells. This results in chemokine release, which even causes more neutrophils to attract to the site of damage and causes even further tissue injury. There's also complement fixation, which causes further chemoattraction of neutrophils and complement fragments using C5a, which is also known as an anaphylaxis toxin. And neutrophils cause even more tissue damage via granular release and oxidative burst. And NK cell activity is also implicated. So in here we have the glomerular stain for IgG deposition by immunofluorescence. We can see antiglomerular basement membrane in the green deposited in a linear pattern on the glomerular basement membrane, causes local activation of the Fc positive cells with complement fixation, neutrophil influx. And here we can see N is neutrophils on glomerulus. So again, we can have a lot of damage. And again, it can be quite fatal. So another example is myasthenia gravis, where we have the formation of antibodies against a host cell surface receptor. So myasthenia gravis is due to autoantibodies targeting acetylcholine receptors. So if you guys have taken any neuromuscular stuff, you'll know that acetylcholine is involved in muscle contraction. And we need it to basically, as our neurons communicate to our muscles at the neuromuscular junction. So myasthenia gravis is an example of an organ specific in the nervous system. So in myasthenia gravis, we have autoantibodies against acetylcholine receptors, or AChR, and these block acetylcholine from binding to the receptors. This means no signal is transmitted to the muscle and causes muscle weakness, and can lead to eventually paralysis. So here we can see a normal one, neuronal impulse we have here in our binding. Here we can see the receptors are diminished and inactivated. Acetylcholine cannot respond. And we can see here that the receptors are essentially internalized and degraded, and essentially we don't have any receptors. So at a normal neuromuscular junction, acetylcholine release from the motor neurons binds to the receptor on the skeletal muscle cells, and this induces muscle contraction. The early stages of myasthenia gravis are due to the blockage of acetylcholine receptors on muscle cells by antibodies generated against this receptor. These anti-acetylcholine receptor antibodies bind to the acetylcholine receptor without activating it. So essentially they block it and they block the binding of our normal acetylcholine and block our neuromuscular transmission. So our anti-acetylcholine antibodies also lead to receptor destruction via type 2 hypersensitivity. This is through antibody binding and the activation of complement and damage. These acetylcholine receptors are internalized and degraded, and then we have progressive muscle weakness. The eyes are often initially affected, then we have difficulty swallowing, food particles can enter the lung and impair breathing, fatal in severe cases. So we can see here the progressive destruction. So type 3 hypersensitive reactions. So some systemic diseases caused by immune complex formation can follow the administration of large quantities of poorly catabolized antigens. So type 3 can be quite serious, and we'll take a look at that. So again, we can see serum sickness and different reactions, so it's quite serious. So type 3 hypersensitive reactions involve something called immune complexes. So antibody-antigen complexes induce damage to tissues through complement fixation and recruitment of neutrophils, macrophages, and mast cells, which cause media release. So here, for example, a type 3 immune complex, we can have manifestations of local arthritis reactions and generalized reactions such as serum sickness, necrotization, vasculitis, glomerulonephritis, rheumatoid arthritis, and systemic lupus erythematosus, which are all quite serious conditions. So serum sickness. So serum sickness occurs when antibody is formed in response to antibodies from other species. So example, horse serum, anti-tetanus antibody used for past immunization. So immune complexes form, and these deposit in capillaries, joints, kidneys, and fix complement and activate phagocytes via Fc receptors, and cause tissue damage and symptoms such as pain, rash, and fever. So serum sickness is usually self-limiting. As immune complexes clear, symptoms resolve. So usually, like if you were injected with something, so you form serum injection, eventually the complexes will go away, so it's not permanent. It'll only be in the system for a certain amount of time. So these kinetics are more rapid if the same antigen is occurred again. So again, this would be more like that. So the secondary response, the symptoms go away in about 1 to 2 days. And serum sickness is an example of a transient immune complex mediated syndrome. So in type 3 hypersensitive reactions, we have an arthritis reaction, which is used to delineate events. So we have immune complexes which form at step 1. A complement activation, including the release of C3A, C4A, C5A, which involves the attraction of mast cells and causes degranulation, including histamine release. You see here, these are mast cells. Then we have neutrophils are also chemoattracted. You see here, through chemokines. are also our complements, which can chemoattract. And then we have the release of our lytic enzymes and tissue damage. So deposition of immune complexes in tissue causes local inflammatory responses, i.e. the Arthur reaction that was on the last slide. So this Arthur reaction is a localized type 3 hypersensitivity response where there's redness and warmth for 1 to 4 hours. Usually has intermediate timing like we saw in the original figure. There can be tissue swelling and edema, which is due to the increased vascular permeability, which is complement mediated. And we also may see hemorrhages, including capillary damage and scarring. So again, here's an example. Locally injected antigen in immune individual with IgG antibody. Local immune complex formation activates the complement. C5a binds and sensitizes the mast cells to respond to the immune complexes. See that here. We have the activation of our Fc gamma receptor 3 on mast cells, which induce their granulation. Here we can see releasing its contents. This results in a local inflammatory response with increased fluid and protein release, phagocytosis, and blood vessel occlusion. And again, we can have capillary damage and scarring as well. So another one is, again, immune complexes can involve, we essentially have bystanders. So even though these complexes are caused by something else, they can get lodged in places that essentially become damaged because of that. So as you can see here, tissues become innocent bystanders. So tissues can be damaged due to immune complex deposits and not because the antibody in the complex is specific for that tissue. So it's not actually attacking this bone here. It's just because this reaction is going on and causing lots of inflammation, which inherently damages stuff. So again, we have all these different things that are being released that can cause damage. So common sites that are prone to immune complex deposition include the joints, kidneys, skin, lung alveolar air spaces. So again, these immune complexes result in complement activation, complement fragments such as C3a and C4a and C5a, which chemoattract neutrophils, and we have damage to proteases and oxidative burst activity. So one of the more serious type 3 hypersensitive reactions are rheumatoid arthritis, where we have a rheumatoid factor for IgG complexes and causing arthritis. So rheumatoid arthritis is a systemic autoimmune disease. Joints are mainly affected due to the immune complex deposition. So these type 3 hypersensitive reactions and Th1 cells are both involved, and involves rheumatoid factor, which is often produced, and these are autoantibodies against IgG, and immune complexes of rheumatoid factor and antibody form and deposit in the joints. So here's an example in the hands, which can be so these are just having intense inflammatory reactions. So in rheumatoid arthritis, or RA, immune complexes lodge in joint cartilage and result in complement activation and the Arthur reactions we talked about. Then we have our small complement fragments, which chemoattract neutrophils. This damage is due to proteases and oxidative burst activity. These macrophages contribute to further damage and cause chronic inflammation results. So there's one of the key therapies for rheumatoid arthritis is blocking TNF-alpha and other stuff like that. So a lot of these become targets for these conditions as well, so keep that in mind. So in rheumatoid arthritis, an unknown trigger sets up the initial focus of inflammation in a synovial membrane located in our joints, attracting leukocytes into the tissue. We have our autoreactive CD4 cells, which activate our macrophages, resulting in the production of pro-inflammatory cytokines, causing sustained inflammation. So again, these cytokines are usually targeted. Cytokines induce the production of MMP, matrix metalloprotease, and rank ligand by fibroblasts. These metalloproteases attack tissues, and there's activation of bone-destroying osteoclasts. So as you may have learned, we have our osteoclasts and osteoblasts. These osteoclasts remove bone, and these get activated unnecessarily, and again, remove bone, which can make it even worse and result in joint destruction. So the synovial membrane inflammation attracts the macrophages and lymphocytes to the site of complex deformation. We have our autoreactive CD4 T helper cells that activate macrophages and release pro-inflammatory cytokines. So for example, IL-1, IL-6, IL-17, and TNF-alpha. Fibroblasts produce matrix metalloproteases, which again cause tissue damage. And these T cells and fibroblasts produce rank L, the activation of osteoclasts, and we have bone destruction alongside this excessive inflammation. So another quite serious condition is systemic lupus erythematosus, or SLE. This is a systemic auto disease due in part to type 3 hypersensitive reactions. This affects multiple organ systems and tissues. The problem is that IG antibodies against our own DNA, RNA, histones, and ribosomes, so you can already see that's not going to be a good time. We have many small immune complexes form, and these deposit in capillary walls, kidneys, and joints. And this is a type 3 reaction that results in chronic inflammatory disease and several progressive conditions in many organs. So these depositive immune complexes end up in the renal glomerulus, which causes renal failure in SLE. So these show a section through the renal glomerulus. So here's A. And see, you can note the clear canals through the glomerulus where deposition immune complexes. There's the thickening glomerular basement membrane. So immune complexes in the kidney membrane cause kidney damage, causing glomerular nephritis. These immune complexes in the synovial membranes leads to joint damage, arthritis, in RA, as well, and SLE. And in some type, we also have some type 2 damage also, where we have anti-red blood cell and anti-platelet antibodies. So immune complexes are most problematic if they are very small and they're less likely to be cleared by our immune system, other cells. They're positively charged, which means they're more likely to stick to negatively charged surfaces. For example, the kidney glomerulus has an anionic basement membrane, so again, it can get clogged there, blocking the kidney. There can be certain inherited complement component defects, which may result in difficulty in clearing immune complexes. So again, such as we had our decay accelerating factor and other cells that are involved in removing complements. So if we have issues in this, we can have overactivation, and this can result in predeposition to type 3. We also can have macrophage phagocytic defects, which also reduce the ability to clear immune complexes. So again, here, essentially summarize. So we have our genetic defects in our complement components, or our macrophages. Our antibody to antigen ratio is too low. Again, positively charged immune complexes are more problematic, and the size, smaller is worse. Whoops. So again, autoimmunity may be induced due to the failure of tolerance mechanism and or environmental factors, such as infection. So we'll get into that now. So essentially, here's our summary figure of what can cause autoimmune. So we have our genetic factors, something exposed in the environment or infection. We have some sort of dysregulation, and this causes autoimmunity. So sometimes when we get to this stage, it can be prevented through different mechanisms like Treg cells and stuff like that. But if it gets past here, we have autoimmunity. So certain genetic factors. So these are examples of certain gene defects that are associated with autoimmunity. So as you mentioned, we have ARRE. We mentioned our CDLA4, which is associated with Graves' disease and type 1 diabetes. So as we remember, CDLA4 inactivates T cells and reduces their effects. So without this, T cells would be overactive. And it's funny because a lot of these things are in autoimmunity, are also essentially used sometimes in other treatments, such as cancer. So for example, here CDLA4 defect is bad, but in some cancer treatments we block CDLA4, so we have more T cells to attack our cancer cells, so we have less energy. But that's for another lecture. So example is FOXP3, our FAST, which is involved in a lot of apoptotic death of our self-reactive B cells and T cells. C1q, which is part of the complement C1q complex. So this results in defective clearance of immune complexes and apoptotic cells. And then we can see here IL-10 receptor A, IL-10 receptor B. So essentially we talked about IL-10 being very important and as an anti-inflammatory, reducing autoimmunity. But if we have defects in the receptors, it won't bind and won't be effective. But yeah, again, go through this if you want more detail. And again, single traits associated with autoimmunity include. And again, you could have multiple and different variations. There's a lot of individuality. So another example is our certain MHC alleles are associated with certain autoimmune diseases. So example, type 1 insulin-dependent diabetes shows an association with certain HLA-DQ beta alleles. These are potentially influencing the ability to present self-peptides to autoreactive T cells and or influencing positive selection in the thymus. So here's a number of different diseases and the HLA allele. So this one is commonly taught is that ankylosing spondylitis is associated with a defect in B27. So essentially, not all of these are guaranteed that if you have this, you'll get the disease, but B27 is quite a high occurrence. So this is if you have back pain or something like that, and they test for this and you're positive, you likely will, there's a high chance of having ankylosing spondylitis. So we can see here with high relative risk. So that's kind of you can see here, if we find this is your relative risk. So that's why this one's reported so much, due to that extremely high relative risk compared to the other ones, as you can see. So again, type 1 diabetes, this one's pretty high with DQ2 and DQ8, as we talked about here. And then again, you can see all the other ones on here. So a lot of these can be tested in genetic counseling and then different risk factors and then lifestyle. So something like diabetes or other ones, you can try and improve your lifestyle to reduce your chances, such as good diet and exercise. So the role of infections. So we mentioned this in that early figure, but we know that certain pathogens can activate self-reactive lymphocytes. And again, sometimes can be preceded by infection. Certain infections are sometimes associated with the onset of autoimmunity. For example, group A streptococcus with rheumatic fever and Coccus B virus with type 1 diabetes. So we can kind of see here, we've seen HLA B27 before, and these ones before. So we saw our DQ2 and DQR. Essentially, it's because these have very close associations with these molecules. So essentially, our immune response to the virus can overlap with their beta cells in our pancreas, and essentially can be called like mimicry. So you essentially mistake a virus antigen for our own antigen. We mentioned this in another lecture. I'm looking at another condition. So infections that cause damage to cell and tissue barriers could expose hidden antigens, like we talked about with damage to the eye, which can cause sympathetic ophthalmia. But we can also have molecular mimicry, where some pathogens may have antigens that cross-react with self-antigens. So, for example, in molecular mimicry, we have the production of cross-reactive antibodies, or T cells. So, example, we can have rheumatic fever, reactive arthritis, and Lyme disease, in comparison to our destruction of the tissue barrier, like you talked about with the eye. And they can these infectious agents can break self-tolerance in several different ways. So, one example here, we have some pathogens have antigens that cross-react. So here we have antibodies against streptococcal cell wall antigens, which can cross-react with antigens on our tissue, causing rheumatic fever. So antigens against certain streptococcal cell wall antigens can cross-react with proteins on heart tissue. The immune response to streptococci produces antibodies specific for varied cell wall types. Cell wall epitopes, some of these shown in yellow in the figure, cross-react with heart proteins, while others shown in blue do not. An epitope in the heart that is structurally similar but not identical to the wall protein can be recognized by the antibody, and autoimmune-mediated heart damage can happen. So it's not a guarantee that if you get this virus, you're going to have this, but it's possible. So here we see they're blue. We produce these blues to this, but they didn't self-react with the heart, but unfortunately the yellow ones did in this scenario. So we can also have bystander T-cell activation. So this infection, where an infection induces co-stimulatory molecules on APCs to present self-antigens, where you have self-reactive T cells in an area that are accidentally activated. So inappropriate induction of MHC class II expression on tissue cells during an infection can facilitate autoimmunity. So for example, thyroid epithelial cells do not usually express class MHC2, but interferon gamma production during infection can induce expression in our thymic epithelial cells, which can then present thyroid peptides to T cells, potentially activating T cells specific for thyroid cell surface receptors, which is not good. So again, this is shown here in this figure. So genetic variants that impair innate immune responses can predispose individuals to developing T-cell mediated chronic inflammatory responses. So one example of a chronic inflammatory disease is Crohn disease. This is a type of inflammatory bowel disease with episodes of severe GI tract inflammation, where we can have granulomas similar to those seen in type 4 hypersensitive reactions that we talked about with tuberculosis. Innate host defenses fail to block gut bacteria from the adaptive immune system. So our NOD2 mutations, which we talked about being a PRR, an intracellular PRR, which they'll have decreased ability to detect gut microbiota-derived material, or MDP. This will result in poor innate responses to gut microbiota, impaired release of antimicrobial peptides, and less containment of gut microbiota. The result is that it allows excess CD4 T cell responses to normal gut microbiota, so essentially attacking bacteria that didn't need to be attacked, causing type 4 hypersensitivity damage to the GI tract. And then here we can see granulomatosis inflammation and Crohn's disease. So Crohn's disease results from a breakdown of normal homeostatic mechanisms that limit inflammatory response to gut microbiota. So again, we have our mucus here, gallbladder cells, our Paneth cells, intestinal epithelial cells. Here we have our dendritic cell activated against bacteria that it shouldn't be, essentially, causing all of these pro-inflammatory cascades when we didn't need it. And there'll be less release of our antimicrobial peptides in this case too. So again, if we have a defective nod, there'll be less antimicrobial peptides. Then it'll get picked up when usually it might be already degraded by these, and then we have an immune response. And again, I hope you guys learned something. So this is a more applicable side of immunology, but again, I recommend going back through any of the lectures to pick up on the pathways. So even here, you can see why this could be an issue. But yeah, I hope you learned something, and see you in the next one.